So, so we're going to look at later on how these 20 amino acids, they actually have a separate catabolic pathway. So different amino acids, they have a different catabolic pathway. And so they will involve these 6 intermediates. Okay, we're going to look at which intermediates actually provide which amino acid catabolisms, and which of these amino acids will enter the citric acid cycle. Okay. Going to clear all drawings. All right. So this is the process of deamination that we had discussed before, where we actually remove this part, right? We remove this amino group here, right here, which is comes from the glutamate. We have this the end product of transamination, which have where we have the high concentration of glutamate, and this part of this amino group will be to remove this amino group is deamination, right? What we call as deamination. And yeah, what's left is carbon skeleton, right? So we have this acetyl-CoA and ketone bodies, right? So we have, as I mentioned just now, this there will be the there will be oxidation. Okay, this two is going to be fully oxidized into carbon dioxide and H2O, and we have glucose, acetyl-CoA, and also ketone bodies. So this is the end product of the carbon skeleton, right? Okay. Okay. So the amino acid catabolism, it's actually linked to the citric acid cycle. Okay, linked to the citric acid cycle. As we have discussed before, okay, in the previous cycle, which is the urea cycle. The urea cycle is actually linked to the citric acid cycle. Okay, so this is what we call, previously they mentioned this as crack bicycle, because, you know, you look at this, it looks like they interconnected between the two cycles, or, you know, the concept of the bicycle. We have 2 tires, right? So this is where the whatever intermediate, okay, this is how the intermediate that involved in the urea cycle. We have the citrulline, arginosuccinate, arginine, and ornithine, and apparently, as you can see here, the arginosuccinate as the intermediate in urea cycle is actually provide to this formation, or to this also provide as to the intermediate as an intermediate in the citric acid cycle. Okay, so these two, they actually interconnected with this one intermediate here, which is arginosuccinate. Okay, and these two keep going as long as they have all the sources, all the intermediates that can provide to the continuous cycle of these two. Okay. Okay, so before we get into details how each amino acids is going to be degraded and what types of intermediate are involved, so we actually can classify. It's easier for us to see these amino acids in 2 categories, which is the according to the class. Okay, so the first one we have the glucogenic amino acids, and the second one we have the ketogenic amino acids. So glucogenic amino acid is based on the amino acid that can be converted into glucose. Okay, and then ketogenic amino acids is, of course, another way around. Okay, the opposite one, which is they actually cannot be converted into the glucose directly. Okay, and so most of these carbon skeletons, they're going to be degraded into the pyruvate or to another intermediate for a 5 carbon intermediates of crop cycle. So this actually can be a precursor for gluconeogenesis. Okay, so in case of glucogenic amino acids, they have this, okay, mainly the amino acids is going to be involved in gluconeogenesis, right? So if you look at here, we have the list, okay, the list of glucogenic amino acids that are capable of forming glucose and glycogen. Okay, so these are all the amino acids that can be used, okay, can be used or can be degraded into the glucose, okay, into the glucose. So if let's say the glucose is accumulated in the body, okay, so then it's going to be converted into the glycogen to be stored, okay, to be stored, and of course it's going to be used whenever the body or other cells needs them. Okay, so these are the all the amino acids that involve in glucogenic amino acids. So of course all this is going to go through the process of gluconeogenesis to provide the glucose, right? Okay, and so these glucogenic amino acids, the carbon skeletons are degraded into this, what we call as they have 4 to 5 carbon intermediates. So we have the pyruvate, oxaloacetate, alpha-ketoglutarate, fumarate, and succinyl-CoA. And okay, so these all these glucogenic amino acids. So which one is going to be degraded into the pyruvate? Which one is going to be degraded into oxaloacetate or fumarate, succinyl-CoA, or alpha-ketoglutarate? So we're going to look at later which of them is going to be degraded into these intermediates. Okay, and then we have also another classification, which is the amino acid. They can be both. Okay, they can be both, whether they are actually glucogenic or ketogenic amino acids. Meaning that these amino acids, they can actually produce the glucose, but they can also produce what we call as ketone bodies. Okay, so this is the 5 amino acids. Okay, 5 amino acids: phenylalanine, tyrosine, isoleucine, tryptophan, and threonine. So these 5, they can be both, whether they want to be the glucogenic or ketogenic amino acids, right? And another classification other than the major one, we also have 2 amino acids, which is strictly ketogenic, meaning that there is no way that these 2 amino acids, they can produce what we call as glucose, because they are strictly ketogenic. So these 2, they can just produce what we call as ketone bodies under the ketosis. Okay, under the ketosis. And so these are the ketogenic amino acids. Okay, ketogenic amino acids. So ketogenic amino acids, so they can be leucine or lysine, which is the strictly ketogenic, or they can also involve these 5 amino acids as well, because these amino acids, they can be both glucogenic or ketogenic amino acids, right? So the carbon skeleton actually will be degraded into acetyl-CoA or acetoacetate. Yeah. So those amino acids that are ketogenic, they are going to be degraded into this, what we call as acetoacetate, and they will be oxidized into ketone bodies. Okay, and this is the 7 amino acids that are going to be degraded entirely, okay, into the acetyl-CoA or acetyl-CoA and ketone bodies in the liver. So we have 2, which is leucine and lysine, right? Leucine and lysine, which is the strictly ketogenic, and we have the another 5, which is listed here, which is also, it can be both ketogenic or glucogenic amino acids. So make up these ketogenic amino acids are 7 in list. Okay, but anyway, you have I think for the glucose, it's quite familiar for you. Because glucose, you have learned about glucose, gluconeogenesis, the formation of the ATP, which involves the oxidative phosphorylation, which is the ETC. I think you have learned about this in a previous, previous class, previous diploma or SPM. I don't know. If you enter matriculations, K matrix, I think you have learned about all these mechanism or pathway or metabolisms of glucose, right? But then in case of ketone bodies, so have you learned about this before, about ketone bodies? Anyone? Have you ever learned or heard about anything that involves ketone bodies before? Okay.
No, doctor.
No. Others? Okay, dua orang je Dua orang je tak pernah dengar. Okay. Okay, so excellent. Okay, 3. Okay, 4. Okay, ramai je. Okay, ramai lah. Okay. Kalau ramai yang dengar, saya tak nak bercerita. Kalau okay, ramai ya. Okay. Kalau ramai yang tak pernah dengar, saya akan explain. Kalau ramai, kalau tak ramai, tak payahlah. Okay. 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 So ketone bodies ni apa dia? So ketone bodies, okay, saya dah lukis tadi. I've draw the one that involve the ketone bodies, right? So ketone bodies ni, it can actually derived. Okay, it actually derived from the protein yang saya cakap tadi. Okay, protein, lepas tu dia ada carbon skeleton. Carbon skeletons, and this carbon skeleton, it can be degraded into glucose, right? CO2, H2O, glucose, and then acetyl-CoA, and then another one is yang ni lah, I just take this one, acetyl-CoA. And acetyl-CoA, it can be actually in both ways, whether this acetoacetate. Sorry, kejap, mana ni eraser? Acetoacetate. Okay, acetoacetate. Okay, acetoacetate. Okay, this acetoacetate later on will be oxidized into the ketone bodies. Okay, so this is the pathway. Okay. So ketone bodies ni apa dia? So it's the chemicals yang dihasilkan. Okay, it's will be produced by the cells if there is no enough insulin in the blood to break down into the fat or energy. Okay, so when is the situation when you don't have enough insulin in the body? So usually what happens if you don't have enough insulin in the body? Do you have any idea? Okay, starvation. Okay, because the glucose, there is no signal, right? There is no signal to actually to convert the glucose into energy, right? Or to into the blood. So that's why it can cause starvation. Okay, good. Okay, another one, another another fact, okay, or consequences if you didn't have enough insulin. Diabetes, yes, that's right. Yes, okay, interesting. Okay, all right. So diabetes, starvation. Okay. Usually, those people. Okay. Ini cuma nak bagi pemahaman so that you can understand further of what is the relationship between the keto bodies, diabetes, starvation dengan all these metabolisms. Sebab if you can get the clear picture, if you can get the big picture, um, this will make you understand and senang nak ingat. Okay, you can easily remember all these facts because you can understand. Okay, the situation or what happened in the metabolisms. So usually, if you okay, we know that the people with diabetes usually those that badan dia besar kan. Okay, it's usually macam tu. Tapi but then mostly orang yang kena diabetes dia cepat jadi berat dia kurang, betul tak? Okay, if I don't know if you know that, but it's actually yes. Those people who get diabetes, okay, they can easily later on they can their weight become drop very fast. Okay, so this is actually what happened. Okay, sebab because because right now what happened to the diabetes patient is that they don't have enough insulin, or insulins become resistant, which is the hormone become haywire somehow, so they cannot give signal to the glucose to make to provide the energy to the glucose to enter the blood and provide the energy. So what happened is that what happens that the ketone bodies, which is actually in the muscle, the ketone bodies yang ada dalam muscle tu, dia akan digunakan instead of the glucose yang ada dalam badan. Okay, so the ketone bodies, bila ketone bodies digunakan untuk to produce the energy, then that's why lah diabetes patient ni dia akan jadi kurus, dia akan jadi kurus sebab because mostly the ketone bodies from the ketone bodies ni akan digunakan sebagai akan degrade as the energy other than fat lah, so that's why jadi kurus. Okay, so that's why if you can understand this relationships, the connection between what happened, I think it's going to be easier for you to understand and remember the about the ketone bodies, about the glucose. Okay, all right. So in the okay, so this lipid droplets. Okay, lipid droplets, and this is hepatocytes, and this is acetoacetate, which actually can which is derived to the formation of the ketone bodies. Okay, so we have this acetyl-CoA, and then we have the citric acid cycle here, which is the citric acid cycle, which will provide the acetyl-CoA, and it will finally produces acetoacetate. Okay, and this acetoacetate will be exported as energy source, okay, to the heart, to the kidney and brain, and other than the ketone bodies, we also have this fatty acids, fatty acids which will be oxidized to this channel to acetoacetate, acetyl-CoA, and it will also provide to the ketone bodies for the energy. All right, so there is another pathway for to provide energy other than carbohydrate or comes from dietary protein. Okay, so that is about the ketone bodies. All right, so if fatty acids and amino acids degradations is very high, then there will be if there is a continuously the process of degradation of fatty acids and amino acids, so there will be a high concentration of ketone bodies produced. Yeah, so this is what we call as ketosis. Yeah, ketosis. What we call the situation, what we call the process, what we call is ketosis, where the fatty acids and amino acids will be degraded, okay, into the formation of ketone bodies. So we call this as ketosis. Ketosis is okay. Ketosis is okay or is normal, but then it can actually potentially cause ketoacidosis. Yang ni kita tak nak. This is what we don't want, ketoacidosis, because ketoacidosis it can actually become it will result in excessive breakdown of fat, can cause the deficiency or inadequate use of carbohydrate. Ini akan menyebabkan, this is will cause the carbohydrate or the source of the main source of carbohydrate or glucose tidak akan digunakan. Okay, tidak akan digunakan. So what happened? The glucose in the blood is going to be accumulate. It's going to be very high. That's why this is related to the diabetes type 1. Diabetes type 1 patient. Okay, because this the glucose, the insulin is really resistant, dah tak boleh bagi signal untuk menukarkan glucose kepada kepada energy. Okay. Ketosis is normal. Okay, ketosis is normal, but then ketoacidosis is dangerous. Okay, dangerous, and it can be fatal if left untreated. So as I said, the blood becomes very acidic and it's very harmful if this ketoacidosis keep going, keep continuously happen in the body. And yeah, so as I said, it's associated with the uncontrolled types of diabetes type 1. All right. So that is the story lah. Okay, the explanation on what is the ketone bodies, and we relate it to the normal the situation, the real life situations about the diabetes. Yeah? Okay. And this is the degradation products of carbon skeletons, which we have is the amino acids. Okay, which amino acids produce which intermediates anyway, because we have discussed earlier that these amino acids, they can be degraded into these intermediates, okay, by the pyruvate, oxaloacetate, fumarate, acetyl-CoA or acetyl-CoA, and these actually intermediates, I'm not sure if you notice that all these actually can be found in the TCA cycle, TCA cycle, and also urea cycle, and even during the gluconeogenesis process itself, because by the presence of these intermediates, then this makes this amino acids catabolisms is very significant to the pathway, to the metabolisms, totally. Okay, so this gradation of products of carbon skeletons, if you look at from here, from this chart, you can see that which amino acids will be degraded into which intermediate. So we have here alanine, cysteine, glycine, serine, and threonine, and tryptophan. So these are very critical amino acids that will provide or to be degraded into pyruvate. Yeah, and then we have asparagine, aspartate. So these actually all these amino acids, when they are degraded into these intermediates, so you can see each of these intermediate is actually what provide to this continuous cycle of TCA cycle. Okay, provide to this continuous provide to the series of the TCA cycle here. So so the yellow one is those that ketogenic amino acids, ketogenic amino acids, and those that in the red, in the red one, in the red shade is the one that's glucogenic amino acids, and this is where, and this is how the oxaloacetate, which is the intermediate of this TCA cycle, which derived from the degradation of this asparagine, aspartate, which is directly from these two amino acids, oxaloacetate. So they can provide to this formation of a phosphomonopyruvate, phosphomonopyruvate, and they can make up the glucose. Okay, the glucose, and the glucose will enter the gluconeogenesis. Okay, so that is the the concept. Okay, and I think, okay, so this one is the same one with this. Okay, just sama saja. Okay, I think this is the same figure. But same figures. Okay, involve the what are the intermediates that involve for from the degradation of each amino acids, whether glucogenic or ketogenic amino acids. Okay, let's look at one by one. We have the pyruvate. So the pyruvate is the actually the product, okay, from the degradation of these six amino acids. Yes, alanine, serine, glycine, cysteine, threonine, and tryptophan. So how actually they are converted to the pyruvates? So the first one is from the amination, okay, from the trans amino acid directly that produce a pyruvate, as we have learned before, through this transamination, right? Because during the transamination, we need to do this deamination to remove the amino group, and we have the product, which is the pyruvate or oxaloacetate, which comes from the aspartate, right? So, but this one for alanine, we have the product is the pyruvate other than glutamate, right? So, still remember this one. This is the transaminase, okay? And then we also have the serine, which will be deamination, or they have a direct deaminations to form pyruvate from this serine dehydratase, which involve the two steps that we have discussed before. Okay, because the amination of serine, it can involve the direct deamination, as we have discussed before. Okay, so it also produces the pyruvate directly. And we have also glycine. Glycine is also can be converted into the pyruvate through the formation of serine as the intermediate at the first place, and then and serine will be and it will be degraded subsequently as the serine degradation. Okay, after this glycine will degrade into serine. Okay, so glycine, before they can be converted into pyruvate, glycine can degrade into the serine. Okay, serine, which involves this serine hydroxymethyltransferase. Okay, they have this enzyme for the formation of the glycine to the serine. And actually, during this conversion from glycine to the serine, they have this PLP. They have this PLP as a cofactor, which will transfer the the methyl group. Okay, will transfer the methyl group. So okay, so from the glycine cleavage, okay, after the glycine is cleavage in the presence of serine hydroxymethyltransferase, this will produce this N5 and 10 methyl tetrahydrofolate. Okay, so after this will be produced, okay, they will actually produce they will receive the one carbon. Okay, they will receive one carbon from the process of the glycine cleavage system, and they will be converted into the serine. Okay, converted into the serine after they receive the carbon metal group. Okay, and then finally the serine will be degraded into the pyruvate as the previous step. Yeah, this one. Okay, so this is the step of how the glycine degraded into pyruvate. And then the next one is we have the cysteine. Okay, so for the cysteine to be converted into the pyruvate, they have to go through this three-step pathway. Okay, which is the first one is the oxidation of the cysteine to the cysteine sulfonate, and the cysteine sulfonate will lose the amino group, okay, by transamination to form this beta sulfonyl pyruvate, and this and the next one is the process of desulfurylation without the presence of the enzyme. Okay, so after the sulfur group of this the beta sulfonyl pyruvate is removed, then we get the pyruvate, and this process does not need the enzyme. Yeah. So this is how the process. So we have the cysteine, and they have the after the oxygen. Okay, after the oxygen is removed, then we have the sulfonate group through this the third carbon, right? And then we have the transamination process, which produce the glutamate, and at the same time also to remove the amino group, okay, from this beta sulfonyl pyruvate. So this is a product. As you can see, after transamination, there is no more upper amino group, right? Okay, from this cysteine sulfonate. So we have the product as beta sulfonyl pyruvate here, and later on we cleave this the sulfur, okay, the sulfur group. We cleave this part from this beta sulfonyl pyruvate. So we cleave this part, but in this case, this is no enzyme involved. It's just directly cleaved, and we get the product as the pyruvate. So this is how the cysteine converted into the pyruvate. Okay, so there is 3 steps: 1, 2, 3. Okay. So the next one is the tryptophan. So tryptophan, a little bit, I think the process is a little bit longer. Okay, it actually involves the 8 steps. Okay, 8 steps from tryptophan to the alpha ketoadipate to get into this product alpha ketoadipate. They have to go through 8 reactions, which is to complete these 8 reactions, they also produce the side product, which is the alanine. Okay, the alanine. As we know, alanine also later on can, via the transamination, they can also provide to the formation of the pyruvate, right? So, but then the tryptophan, if they want to go through to the end product, which is the pyruvate, so they have to go through the 8 reactions here up to alpha ketoadipate, and then they have to go through another 6 reactions. Yeah, 6 reactions to the formation of the pyruvates. They have these 2-acetyl-CoA, okay, 2-acetyl-CoA, and also the 2 carbon dioxides, right? And the next one is the threonine. Okay, threonine. Okay, for the threonine, it can be converted into the acetyl-CoA and glycine. Okay, and we know that the glycine will be converted into serine, and then serine will be converted into the pyruvate. Okay, lastly, but for threonine, it's not like a direct one-way formation of the pyruvate. So it has to be converted into the glycine at first. Okay, and then the glycine enter the serine, and the serine into the pyruvate. So, but then the threonine, it can be actually both, whether it is ketogenic or glucogenic. So the part that we have discussed, the threonine go into glycine, serine, and the pyruvate. So that one is the part where the threonine enter the glucogenic pathway. But then if let's say the ketogenic, for the ketogenic part or ketogenic pathway, so threonine have to go, they will be converted into acetyl-CoA and then acetoacetate, and acetoacetate can be the ketone bodies, right? And yeah, so this is the part where the conversion of the threonine to the pyruvate. Okay, and then glycine to the serine. Yeah, so we have take a look on the how the formation of the glycine to the serine, right, before in the previous slide. So it's actually repeating of this process until the end we get the pyruvate. All right. So so here is the the summary. Okay, the summary of the those amino acids that degraded into the pyruvate. So where does the process starts? So let's say the threonine, it has to go to the formation of the glycine and the serine have to and end up with the pyruvate in this pathway. All right, and then the glycine starting from here, it go through this. Yeah, so it's just repeating the formation of this methyl group, and then to the serine, and we get the pyruvate. And this is the alanine from this pathway. It has to go through the transamination and get pyruvate, and this is the cysteine. So this is the the combinations of all the amino acids degradations into the pyruvate. Yeah, so it can be this is a long pathway starting from threonine, and on its way to this pyruvate, it can be converted into glycine and serine, and it get the pyruvate. Okay. Yeah. So this is just, I think this is just a similar one. Okay, similar one with this figure, but this is quite. This is more details together with the enzymes involved during the process, and also include the structure, what happened, what are the changes involved during the conversions from one intermediate to the next one. All right. So here is the summary. Okay, a table which show the summary, the breakdown of amino acid to pyruvate. So what are the amino acids? What are the important reactions that involve during the process or during the conversions from each intermediate to the next one? And what are the enzymes involved during the process? And what, of course, the product is a pyruvate lah. Okay, because these all involve the degradations of this amino acid to the pyruvate. Okay. Okay. Again, this is the. I think this is the similar figures that I showed you before. Just to show the what are the amino acids, where they are degraded, and what the intermediates that produced during the degradation of these amino acids. Okay, let's take a look on the some inborn errors in metabolisms. Okay, so here what we have discussed from this topic is just involve the degradations of the amino acids to the pyruvate. Okay, so we know that we have 20 amino acids, and different amino acids, they will be degraded into different intermediate, right? Alpha ketoglutarate, skeleton-CoA, fumarate, salicylated, and so on. But then what we have discussed before, yang panjang-panjang tu, which amino acid will be degraded, is just those that will be degraded into pyruvate. Itu sahaja lah. Okay, but then if you're interested to know more about how each of these will be degraded into this intermediate, you can dig in. Okay, you can find out all those in any textbook or any any reference book or journals if you're interested in. But I just discussed on the part of the pyruvate. Okay, but of course you need to know what are the amino acids that degraded into which intermediates and so on. Okay. because because we have many amino acids. So if I need to, to discuss each one of them, it's going to take a longer time, and Um, so inborn errors in metabolisms. So if let's say so this is more towards the what happened to the metabolisms if there is the abnormalities involved in protein metabolism. So this is inborn errors in metabolisms, which means that it caused by the gene itself. So meaning if you get uh, if someone get this the disease starts from the gene itself, so there is no cure lah. Okay, there is no cure because it's inborn errors. It comes from the gene itself. So basically, if someone get this inborn errors kind of disease, they just have to take precaution. They just have to control the diets, just to control the daripada disease jadi lebih teruk lah. Okay, if that is the inborn errors disease. So some of it, okay, for example, that we have here, inborn errors in metabolisms, we have the tyrosine. Okay, so we have the tyrosine-related disorders where because of this enzyme, okay, the enzymes that should degrade the tyrosine to this or phenylalanine to the tyrosine is absent in this case. So we have so many pathways that actually can be errors because of the abnormalities or absence of the enzymes. So this way is the Hawkins-Sinuria disease. Okay, and if let's say there is another disease that related to the tyrosine as well, which is the tyrosinemia. It can be type 2 and type 3, and and this is to the pigmentation, related to the pigmentation, and also alkaptonuria, which is if let's say homogeneity will be oxidized, they can actually lead to this disease, what we call as alkaptonuria. Okay, and this is another disease which is tyrosinemia. So you can see we have actually a lot of possibilities that this tyrosine, it can be some errors or some disorders in the pathway of degradation of the tyrosine. Okay, because the tyrosine, the fate of tyrosine, actually it has to be degraded into acetoacetate or fumarate, which is this fumarate will enter the CA cycle, right? So but then on the way towards this the fumarate as the product from the tyrosine, degradation of the tyrosine, it can be anything. It can happen all these things because of the some errors caused by the gene and the presence of the enzyme. Okay, so thank God that hopefully none of us have this possibilities. Okay, and one of it, as I said, alkaptonuria is related to this oxidation of homogeneity, where this homogeneity because of the absence of this homogeneity oxidase activity, so the homogeneity will be oxidized. It will be oxidized. So this can cause the urine of this patient become darker because it's easily oxidized. Okay, so this is normal urine, but then for patient with the alkaptonuria, they're going to have this black urine. Okay, and then then they have this formation of the spots like this, the dark spot at the area of the eyes, white eyes. Okay, this is what we call as sclera. This when this enzyme homogenization oxidase activity is the absence, so this makes this homogenization will be easily oxidized in the air when they exposed to the air. Okay, and it will become a highly colored polymer lah macam ni. Okay, dark color urine, and they have sclera at the dekat mata. Okay, and this is another inborn errors as well, which involve the tyrosinemia. Tyrosinemia is the absence of the this enzyme fumarate acetase. Okay, those that have this tyrosinemia, so they have this inability of these enzymes here, yeah, fumarate acetase here. Okay, so it can cause the enzyme defect in tyrosinemia type 1. And we have also albinism, okay, the absence of melanin pigment. And actually, this tyrosinase can be in many pathways. In just small amino acid like tyrosine, since it has a many pathway, okay, many enzyme involved. So at this side is the part where the what we call as umolanins are more towards the the presence if I'm not mistaken lah. Okay, if I'm not mistaken, more towards the melanins or pigment to the part of the hair, to the hair, and which is become darker. Okay, but then for fewer melanins, formation of fewer melanins, the hair will become macam blonde kan, tu ke blonde, rambut yang yang kuning tu, okay, orange tu, ni dia sebab dia terlebih this pigment of your melanins, yeah. Okay, and okay, more on the albinisms. Okay, albinisms. So we even have the albino even in animals. Okay, and okay, so this is another disease which is maple syrup urine disease. Okay, maple syrup urine disease ni because of the lack of the branching the hydrogenase activities. So what happened is they are going to have the accumulations of this compound, what we call as 2,4-dinitrophenyl hydrogen derivative. So this one, the urine will smells like maple syrup, and in this case, the patient will have the mental retardations. Okay, unless they have to take they have to take this valine-free thyroid powder, okay, or they have to take less isoleucine, leucine, and valine in the diet. Okay, so because they because they have the inability to process these alpha keto acids. Okay. And we have the tyrosinemia, which has we already discussed before, but tyrosinemia. Okay, and the what are the cause and what are the what is the defect or abnormalities that involve if you get the tyrosinemia. Okay, so I think that's all for the amino acid catabolism. So any questions plus about this? We dah pukul 10 dah. We memang panjang lah topik ni ya. So we have just 2 lectures, 2 weeks just for this amino acid catabolisms. Okay, tu tak masuk lagi tu degradation for each amino acid je tadi. So I just cover only those degradation of amino acid into just pyruvate. Okay, right. So any questions? Tak ada. From the chat tak ada. So I think I dah letakkan this lecture on the Putra Blast about these amino acid catabolisms. Okay. So yeah, I think for the previous attendance, I tak letak dekat Putra Blast. So so nanti you sign 2 kali, okay? Dekat Putra Blast. Last Monday dan juga hari ni. Any other questions? Ada tak? Ada Kalau tak ada, then I will stop here lah. Okay, I will end the session here. Okay, so this afternoon, group B, please come earlier. Kalau you nak balik awal, nak buka puasa awal, you datanglah awal. Okay, pukul 2 kita start eksperimen. Okay, group B.
Okay, doktor.
Okay,
Okay, doktor.
Okay, doktor.
Alright. Okay. So I will end the session.
Thank you, doktor. Stay safe.
Bye.
Thank you, doktor.
Thank you, Doctor.